Hello, everyone. Thank you very much to the organizer for the invitation to give this talk today. My name is Maria Maun, and I am a wheat pathologist at Oklahoma State University. For today's talk, I am going to share with you part of my work during my PhD studies at North Dakota State University to identify sources of resistance and also resistant genes in tetraploid durum wheat. This is the outline of my talk. I will start by giving a brief introduction on the three rust diseases, leaf rust, stripe rust, and stem rust. And then what I did to identify resistant genes in the NDSU elite durum wheat genotypes. And then what I did to bring in new sources of resistance from worldwide germplasm collection of durum wheat. Although my work is done on tetraploid durum wheat, however, we know that the durum wheat shares the A and the B genome with the hexaploid common wheat. So the resistant genes that are identified in durum can also be transferred and used in common wheat. Starting with the first disease, which is leaf rust, caused by the fungus Puccinia triticina. It is an obligate biotroph, which means it requires a primary host, a living tissue to survive. And the disease is shown here as this circular to oval in shape pustules that are brown, orange to brown in color. This fungus is heteroecious, which means it requires a primary host, which is wheat, to undergo its asexual cycle, and an alternate host, which is talectrum, to undergo its sexual cycle. However, in the US and in many countries, the alternate host is resistant. So most of what we see is a clonal population where there is no sexual recombination. Stripe rust caused by the fungus Puccinia striformis form specialis tritici. It shows here as these elongated yellow stripes on the leaf surface. So it's named stripe rust or yellow rust. This is a very common disease now. Basically, is present everywhere wheat is grown. And in this heat map, I am showing you the disease incidence with a warmer color corresponding to higher disease incidence that are present in countries like the United States, China, and Australia. Under high disease pressure, this disease can cause up to 45% of yield loss. Stem rust caused by the fungus Puccinia graminis form specialis tritici is a very dangerous disease throughout wheat throughout the history, and it caused several epidemics. And in the US, and since the 1950s, this disease was under control thanks to the eradication of berberis, which is the alternate host of this fungus, and the use of resistant varieties. However, in 1999, a new race appeared in Uganda called UG99, and it has virulence on many of the stem rust resistant genes that are deployed in global wheat varieties, making 80 to 90% of wheat varieties susceptible to this race. This race rapidly spread to cover all East Africa, going all the way to the Middle East, Yemen, and Iran. And here are the different races of the UG99 lineage that have some virulence variation on the deployed resistant genes in wheat varieties. However, the current or the recent epidemics of stem rust in Europe, in Asia, and in South America were caused by different races than the UG99. So this shows that we should worry not only about the UG99 races, but also other races that appeared in the region that has also several combination of virulence to stem rust resistant genes. In some countries, like in Siberia, Ethiopia, And in Spain, there were evidence of sexual recombination where scientists found this Asia formation on the leaf surface of barberry, which shows that this fungus is actually undergoing its sexual cycle. The best management approach to manage rust diseases is to deploy resistant varieties. And for wheat rust resistance, there are two types of resistance. Seedling or all-stage resistance, which is complete type of resistance. It is qualitative. It is controlled by one or few genes, and it is caused by caused by hypersensitive response. It is race-specific, however, and it is short-lived. So relying only on this type of resistance will create a selection pressure on the pathogen that can evolve rapidly to this type of resistance. And the resistant varieties in the past can turn to be susceptible. The second type of resistance is adult plant resistance, or APR. It is a quantitative type of resistance. It is has a diverse resistance mechanism that provide a partial type of resistance. And resistance in many cases is to multiple races and also to multiple pathogens. And this type of resistance is more durable. So the ideal in here is to pyramid two types of resistance in one varieties so that we can have a protection both at seedling and adult plant stage and also have a durable type of resistance. Starting with the first part of my talk, which is on rust resistance in elite durum wheat. So I used germplasm from the NDSU, or the North Dakota State University durum wheat breeding program, led by Dr. Elias, who's the durum wheat breeder in there. And I collaborated with Dr. Matraus from the USDA Cereal Disease Lab in the University of Minnesota to perform the stem rust evaluation. And the evaluation for these three rust diseases was done at seedling stage. So the genotypes are 248 breeding lines and cultivars from the Uniform Regional Durum Nursery from 1997 to 2014. This germplasm was genotyped using the Illumina 90K wheat array that generated around 11,000 SNPs. And the phenotyping was done for for three diseases: leaf rust, ripe rust, and stem rust at seedling stage. For leaf rust, and the evaluation at seedling stage was done using 7 races. The first race is an adapted race to bread wheat, where all the lines were resistant to this race. While when we use the adapted races to durum wheat, we reach up to 100% of susceptible lines in the NDSU durum. So this shows here that there is a distinction between the races that are adapted to durum versus the races that are adapted to durum wheat in terms of virulence on durum wheat lines. Using the genotypic data for these three races where we have, or three isolates where we have phenotypic variation and the molecular markers and performing the GWAS, I was able to identify 2 loci that are possibly LR52 and 46, and some novel loci like the one on chromosome 6BL and chromosome 2AS. For stripe rust, we had better scenarios where we have around 45% of the lines were highly resistant to these three races. But the problem is that this resistance was mainly conferred by 2 loci, one on chromosome 6BL, 5BL, and the other one on chromosome 7BL. We don't know stripe rust resistant genes that are present in these chromosomal locations. However, by looking at the literature, I found a breeding lines from Canada that carries a Q12 on chromosome 5BL and 7BL for stripe rust response that are within the genomic region of my identified Q12. So I believe they are possibly the same. For stem rust, we did the evaluation using 3 UG99 races. One race, TKTTF, it's called also the Digallo race that caused the epidemics in Ethiopia in 2013. TRTTF and JRCQC, they are also from Africa, and they are different than the UG99. So we identified mainly 3 genes, SR13, SR8155B1, which is close to SR8, and SR4AL, which is close to the stem rust resistant gene SR7, or it can be, so it can be even an allele of SR7 or tightly linked to SR7. But we also identified some novel loci that are highlighted here with these red circles that that requires further identification and characterization. I developed 4 CASP markers from the SNP markers that I identify that are associated or linked to this stem rust resistant genes that have good accuracy in identifying the genes. And all of these 3 genes, as shown in this figure, are present in high frequency. So these CASP markers, or competitive allele-specific PCR markers, can be used in marker-assisted selection to keep this 3 combination of stem rust resistant genes in future release varieties. In summary, for this part, we identified elite genotypes that were highly susceptible to isolates that are adapted to durum. For stripe rust, we have good level of resistance, but the resistance was conferred by only 2 loci, one on chromosome 5BL and the other one on 7BL. For stem rust, we have more than 80% of resistance for most of the races, and the resistance was conferred by this 3 gene mainly, and also some novel loci. And marker-assisted selection, or markers were developed for marker-assisted selection specifically for these 3 genes, so that we can keep them in future varieties. The second project was to identify novel sources of resistance that are not present in current modern breeding programs, whether it is durum or common wheat. So one way to look at that, I hypothesized that worldwide germplasm collections that are comprised of land races could identify even underutilized or novel resistant genes that we can transfer to adapted durum varieties, especially that in some cases, like for the leaf rust, where we have complete absence of resistant gene to leaf rust to the races that are present in southern US and in Mexico, and also these races are present in the Mediterranean region. So to do that, I use the USDA National Small Grain Collection of durum wheat that was evaluated for leaf rust and stripe rust. And this is a publicly available germplasm. So I was able to share it with other scientists in the US and in Mexico to do the evaluation. So this around 500 lines, most of them or half of them are land races, and the remaining are cultivars and breeding lines from around 70 countries. And the test was done both at seedling and adult plant stages. So for leaf rust, I was able to work with Dr. James Colmer in Minnesota, And for leaf rust evaluation at adult plant stage, I work with the CIMMYT durum wheat breeder, Dr. Karim Amar. And for stripe rust, I work with Dr. Chen from Washington State, or USDA or Pullman, Washington, where, as we know, Washington is a hotspot of stripe rust in the US. So we have um, several races in the region, and also the environment is conducive for stripe rust. For leaf rust, the GWAS allow the identification of 37 loci that are associated with leaf rust response. 14 of them were possibly novel that are shown here with the stars. And we know that the GWAS has a limitation when it comes to rare alleles. So we use the complementary approach, which is based on biparental population mapping, to identify leaf rust resistant genes to this 5 biparental population. And the race that I used for the mapping was the race that is highly virulent on the durum, which is race BBQD from California. So I did inheritance study, and I found that the leaf rust resistant genes was conferred by a single leaf rust resistant gene. So bulk segregant analysis was the rapid and a very efficient approach in this case that allowed the identification of the chromosomal region. Most of these resistance genes are possibly novel, and that was not surprising because most of the genotypes are non-races. Then I use the SNP markers that were identified from the GWAS to further delimit the genomic region of the identified leaf rust resistant genes that are shown here with the arrows. So this allowed to identify competitive allele specific PCR markers from that are linked to these resistant genes, and that can be used directly to facilitate the transfer of these resistant genes into adapted durum wheat varieties. The population RASI by PI 1951 was a very interesting population because it carried both resistance. The PI 1951 was resistance to both leaf rust and stripe rust. So the mapping was done for both leaf rust to these different races, and we were able to identify a novel leaf rust resistant gene on chromosome 4AS, and also a novel leaf rust resistant gene on chromosome 7AS that was mapped in a genomic region where no previously identified stem rust resistant genes were found in this location in both durum and common wheat. And this gene was effective to many races, whether from North America, from the UG99 race, and also races from Africa, and also here in the field in the stem rust nursery in Debrecen, Minnesota, Ethiopia. And here was a minor effect QTL that was race specific on chromosome 4A. This germplasm was also evaluated at seedling stage to 7 races from Washington, and the GWAS allowed the identification of more than 50 loci. Half of them are possibly novel, as they were not known to be present close to known stem rust or stripe rust resistant genes. The ones that are highlighted here in blue were previously known to be present whether in durum or in emmer wheat. And at adult plant stage, the evaluation was done for 2 years and in 2 locations in Washington, and that allowed the identification of adult plant resistant sources that are in here. These are the accessions where you can see that the infection type and the severity at seedling stage was higher than the adult plant stage reaction. So this is an indication of an adult plant resistance. In summary, novel rust-resistant sources were identified in the USDA National Small Grain Collection. Both type of resistant sources were identified at adult plant stage and seedling stage, or all stage. And the resistance sources were introgressed into adapted NDSU durum wheat lines with the help of markers the cusp markers that can be used for marker-assisted selection. With that, I come to the end of my talk, and I would like to acknowledge the people who contributed to this work, my funding sources, and I would be happy to take any questions that you may have. Thank you.